Hey everyone. In this chapter, I'm going to tackle a thorny issue in the neurobiology of learning and memory. The problem is that so far I've described LTP, and by inference, learning and memory, in stages that actually cannot account for plasticity that lasts for weeks or even up to a lifetime. Yet, we saw that LTP can last for at least a year, which was actually the end of the experiment, so hypothetically it would have gone on longer. And we know that memories can last a lifetime. So we spent a few chapters working on the initial stage of LTP induction. I described this stage primarily in terms of activation of kinases that set into motion some reversible post-translational modifications of proteins. I described those kinases as kind of enduring, but we know that that's only really in the fairly short term across perhaps hours. So accordingly, overall, this initial induction stage is a transient stage of synaptic modification involving those kinases. Then I described the mechanisms that induce local protein synthesis at activated spines from pre-existing mRNAs. Now, those lasted a little longer than the induction stage that simply required reversible phosphorylations. However, this stage too was seen to eventually break down if not supported by genomic cascades. And then there are the genomic cascades of signaling that mediate hypothetically more permanent changes in dendritic spines. However, in this chapter, I'm going to ask you to consider that even those changes may not be adequate to encode enduring, self-replicating stabilization of memories that can last a lifetime in the face of constant biological turnover. It's a commonly observed phenomenon in biology that systems constantly tend back toward the mean or fall back to baseline states. So why should it be any different with synaptic function and spine profiles? Clearly, we're still missing something. We need something more to explain enduring potentiation that can last beyond the changes I've described thus far. And I do have a candidate mechanism for persistent and perhaps even lifelong potentiation. And would you believe? It's a kinase. I'm going to introduce a very special kinase mechanism known as protein kinase M-zeta, which you can see if you look closely at the dendrite and spine on the diagram on this slide. The kinase has some unique properties among kinases that make it an excellent candidate for a memory molecule, and it's the topic of this chapter. So let's dig in and let me introduce you to the protein kinase M-zeta. So let's look a little more closely at this idea that molecular turnover is a problem for the mechanisms we've seen so far. Clearly, the initial reversible stage of LTP, when kinases phosphorylate second messengers and receptors to potentiate the kinase, is subject to molecular turnover that would depotentiate the synapse back to its naive state. The phosphorylation-induced increases in single-channel conductance and membrane expression of AMPA receptors, for example, can both be reversed by simple dephosphorylation. This is illustrated clearly for the case of membrane expression in the diagram on this slide. Stimulation-induced increases in AMPA receptor expression in the membrane will eventually be reversed, and the membrane expression will be returned to basal levels by endocytosis. But I want you to notice something else in the diagram. The internalized AMPA receptors in the third figure from the left look like they're being broken down, and indeed, it's true. There's an intimate coupling between internalization of receptors, ion channels, and other membrane-bound proteins and enzymatic breakdown of those proteins. It's not always the destiny of endocytosed membrane mechanisms that they are degraded, but it often is the case. Thus, there really does seem to be a regression toward the mean, or in other words, a return to basal conditions is favored. Even in the case of new proteins and structures from genomic cascades of signaling, those proteins are continuously broken down. And even if they were replaced in a static, enduring system, their replacements would not be automatically phosphorylated or activated by whatever means. So for example, your AMPA receptors are phosphorylated and inserted into the membrane, and then stabilized in the membrane. And we have changes in scaffolding proteins and all the rest while that happens. But eventually, they're endocytosed back in. And when they are endocytosed back in, often they are degraded. And when they're degraded, well, perhaps we could replace them by making more AMPA receptors. Those AMPA receptors that are newly synthesized are not phosphorylated. There's no kinase activity that endures at this point in time that we've seen so far. So how would we maintain the potentiated spine? It's going to collapse back to its naive state, at least in terms of the mechanisms that we've seen so far. So we see that LTP and memories can endure for weeks or a year or even a lifetime. So how can this be reconciled with constant molecular turnover and a regression toward the mean? How can this be reconciled with the fact that new proteins would not be activated? How can potentiation of synapses be maintained in the very long term? Sir Francis Crick, Nobel laureate and one of the 3 people responsible for identifying the structure of DNA, summed up this conundrum very nicely. He wrote, "Almost all the molecules in our bodies, with the exception of DNA, the genetic material, turn over in a matter of days, weeks, or at the most a few months. How then is memory stored in the brain so that its trace is relatively immune to molecular turnover? The molecules in synapses interact in such a way that they can be replaced with new material, one at a time, without altering the overall state of the structure." Now, to Francis Crick, this was a puzzle. And to you and me, in just a few minutes, it will not be. And again, I will assert that the answer lies in a kinase mechanism, but a very tricky kinase mechanism that has the power to self-replicate and maintain the potentiation in the face of constant molecular turnover. And there you go again. I've given away the punchline too early. But self-replication of this kinase seems to be the key. Let's show you what I mean. Now, we previously saw that GluA1 homotetramers are transiently inserted into the postsynaptic membrane after tetanic stimulation. Protein kinase A phosphorylates serine 845 to cause exocytosis into the extrasynaptic region of the spine. And then protein kinase C phosphorylates the GluA1 subunits at serines 818 to anchor the receptor to the postsynaptic density, and at 831 to increase conductance of ions through the receptor. But then these receptors are endocytosed and replaced with GluA2 containing AMPA receptor heterotetramers that form the stable expression of AMPA receptors in the membrane. Now, it turns out that protein kinase C continues to play a role in the delivery of the more permanent expression of AMPA receptors. But there'd be a problem if we were depending upon the classical or conventional form of protein kinase C that I described back in chapter 4, when I described the structure of protein kinase C with a regulatory region that possessed a tail that contained a pseudo-substrate to silence enzymatic activity of protein kinase C in between stimulations. In between stimulations, obviously, conventional protein kinase C would not be able to continue to maintain the stability of GluA2 containing AMPA receptors in the membrane, because protein kinase C will eventually become inactive again. So with the constant turnover that I've been talking about, there isn't a mechanism in conventional protein kinase C to maintain the phosphorylation and therefore the insertion of GluA2 containing heterotetramers into the membrane. So we clearly need another mechanism. Perhaps it might be another form of kinase related to protein kinase C. You see, protein kinase C is actually a family of kinases that differ slightly in their structure and function. In the diagram at the bottom right, you can see that all 9 members of the family have the same kinase domain, shown in blue at the right of the diagram. However, there are significant differences in the regulatory regions. Overall, the 9 members of the family belong to 3 main groups. There are the conventional isoforms of protein kinase C, known as protein kinase C alpha, beta, and gamma. They have the regulatory region that I described in chapter 4. Excuse me. With the calcium binding and diacylglycerol binding regions and the pseudosubstrate tail. I know that we remember this structure, at least a little bit, right? Then there are the novel isoforms, sleepy, sneezy, dopey, and grumpy. No, wait, those are Snow White's dwarves. Okay, just making certain everyone's still following. Yeah. Okay, the novel isoforms are protein kinase C delta, epsilon, eta, and theta. And the atypical isoforms are known as protein kinase C zeta and protein kinase C iota lambda. But there's one more relative, and it's on this one that I will especially focus. It's known as protein kinase M zeta. You should be able to see that it does not possess a regulatory subunit at all. This is going to be very important when we consider the need for a protein kinase zeta that can stabilize AMPA receptors in the membrane on an ongoing basis without the need for reactivation of newly expressed proteins in the face of constant molecular turnover. But that's only part of the story. Let's explore how protein kinase M zeta actually works. Another thing that I want to note about protein kinase M zeta is that it is specific for the nervous system. In other words, you have kinases in cells all over your body. Cells of the kidney and the liver need to regulate protein function, and they, like the brain, do so in part with kinases and phosphatases, phosphorylating and dephosphorylating target proteins. So in the kidney and liver, and all other organs of the body, we find protein kinase A and CAM kinase 2 and protein kinase C and MAP kinase and so on. However, protein kinase M-zeta is not found in the kidney or liver or anywhere else in the body other than the brain and spinal cord cells, as shown in this Western blot, showing protein kinase M-zeta protein from homogenates of kidney in comparison with homogenates of other brain regions. And this makes sense because there are no other cells in the body that need to create permanent changes in their structures and functions that would allow them to store a memory for a lifetime. Yes, some cells do exhibit some plasticity, but it's minor and transient compared to the plasticity exercised by the brain and spinal cord. So as I build the case that protein kinase M-zeta endows the nervous system cells with a unique ability to store memories for a lifetime, you should be able to see that it is perfectly appropriate that cells outside of the nervous system don't need this perpetual memory molecule, and in fact, wouldn't do very well if they were making permanent changes in their function. So let's examine what it is that protein kinase M-zeta actually does. As I suggested on the slide where I revealed the structure of protein kinase M-zeta, the kinase domain is the same as it is in isoforms, in other isoforms of protein kinase C. That means the consensus sequences it recognizes are the same, and as long as it is present, it will exercise the same actions as the isoforms of protein kinase C. One of the actions of conventional protein kinase C is insertion of GLUA1 homotetramers into the membrane shortly after tetanic or theta burst stimulation. Another may be to initially insert GLUA2 containing heterotetramers into the membrane after stimulation. However, the activation of conventional isoforms of protein kinase C may be somewhat transient, so it's not clear to me that the conventional protein kinase C is even still activated at the time when the homotetramers are withdrawn and replaced by the GLUA2 heterotetramers. So there may actually be a mismatch in the time course of conventional protein kinase C activation and GLUA2 heterotetramer insertion into the membrane. But perhaps protein kinase M-zeta comes to the rescue. Protein kinase M-zeta is available within about 10 minutes after tetanic or theta burst stimulation, and endures well beyond the time point when the AMPA receptors would be swapped out for the heterotetramers. So the early availability of protein kinase M-zeta suggests that it could participate in the insertion of the GLUA1 homotetramers that allow calcium influx. And the enduring availability of protein kinase M-zeta suggests that it may contribute to the cycling in of the heterotetrameric AMPA receptors. And did you notice that I did not say that protein kinase M-zeta becomes activated? I said that it becomes available. We've seen activation mechanisms for protein kinase A with cyclic AMPs binding to and dissociating the regulatory subunits. We've seen activation mechanisms for CAM kinases with binding of calcium calmodulin. And we've seen activation of protein kinase C with calciums and diacylglycerol binding. And of course, there are additional mechanisms of activation for myriad other kinases like MAP kinase, ERK kinase, and so on. But in each of these kinase activation mechanisms, the signal activates the kinase by removing the regulatory domain from the catalytic domain. Protein kinase M-zeta, again, has no regulatory subunit. There's no steric hindrance of the catalytic subunit. So this kinase does not need to be activated. It merely needs to be translated from local, constitutively available messenger RNA. And that's shown in the diagram. An influx of calcium activates an inducing mechanism that promotes induction of the expression of protein kinase M-zeta. But then part of the magic will be looking at how protein kinase M-zeta then keeps on going and going and going in a cycle of self-perpetuation. So if we want to understand the availability of protein kinase M-zeta, we need to take a look at the control of the expression of the gene that encodes the protein and the mechanisms of translation of the mRNA. So let's start with the gene expression itself, that is, the transcription of the gene into messenger RNA. Now, protein kinase C-zeta and protein kinase M-zeta are actually both encoded by the same gene, the PRKCZ gene. However, this gene encodes two different mRNA transcripts. One of those transcripts encodes the atypical protein kinase C isoform, protein kinase C zeta, with its protein kinase C catalytic domain, as we saw a few slides back. This is a full-length transcript from the gene, and the resulting protein has the standard protein kinase C catalytic region and the regulatory subunit. The regulatory subunit is a little different from the conventional protein kinase C isoforms that have their calcium and diacylglycerol binding domains, so it's one of the atypical isoforms that is activated by a somewhat different set of mechanisms. But nevertheless, it is regulated by cellular activity and follows the same kind of rules overall as conventional protein kinases, with stimulation-induced activation. The other mRNA transcript encodes protein kinase M-zeta. This is a shortened or truncated form of the mRNA transcript that encodes a protein that has the catalytic region, but no regulatory region, as you can see again in the cartoon of the protein. The difference between regulated and unregulated protein outcome is very important in what's going to happen with the kinase activities of protein kinase C-zeta and protein kinase M-zeta. So how do these different transcripts both get encoded by the one PRKCZ gene? The answer derives from the fact that transcription of a gene is turned on by proteins known as transcription factors binding to a region of the DNA known as the promoter region. We've already seen this previously, where Krebs bound to the DNA and induced gene expression through its actions at that binding site. Well, it turns out that the PRKCZ gene actually has two promoter regions. One of them induces transcription of the full transcript to express the full-length mRNA. The other is an internal promoter site that encodes a truncated protein that's missing the regulatory subunit. This latter version is protein kinase M-zeta. And I think I'd better move on to the next slide to actually give you a visual of this before I say anything more. So here we have it. You can see at the far left end of this cartoon of a gene sequence, again, the best I can do in terms of computer animation, that the promoter 1 region, shown in green, activates expression of the full-length gene, including the regulatory and catalytic domains that are shown in blue. However, if the internal promoter 2 is activated by an appropriate transcription factor, the transcription proceeds to make a messenger RNA that builds only the free catalytic domain. Now, I already told you that protein kinase M-zeta protein is available to start phosphorylating spine proteins within about 10 minutes. So clearly, the local expression of protein kinase M-zeta is not dependent upon genomic cascades. We know that those cascades take hours. Therefore, protein kinase M-zeta must be one of those constitutively expressed proteins, and it absolutely is. Protein kinase M-zeta messenger RNA is synthesized on an ongoing basis and trafficked to all parts of the soma and dendritic arbor. Of course, not to the axons, because you remember that the axons do not accept transport of mRNA or engage in translational activities. The protein kinase M-zeta messenger RNA is trafficked to all parts of the soma and dendritic arbor, kind of a lot like what we saw with ARC messenger RNA. But if the protein kinase M-zeta mRNA transcript is so freely available, it also needs to be protected so it's not translated willy-nilly all over the cell at unactivated spines in addition to the more appropriate stimulated spines. All right, let's pause for a moment. Do people still say willy-nilly? Or is that as outdated and arcane as like oopsie-daisy? All right, I digress. The protein kinase M-zeta transcript is protected from inappropriate translation by a translational repressor protein. That repressor will have to be removed in order to initiate translation at stimulated spines. And here's a key point. The protein kinase M-zeta itself interacts with other proteins to remove the repressor protein. In this manner, protein kinase M-zeta is actually self-replicating. The presence of protein kinase M-zeta leads to initial expression of the transcript so that you have additional protein kinase M-zeta. Let's show you what I mean by all of that on the next slide. Okay, here we go. This diagram fills in a couple of the details. First of all, protein kinase M-zeta messenger RNA is constantly transcribed in the nucleus and trafficked out to the dendrites. However, the protein kinase M-zeta messenger RNA is translationally repressed. Therefore, even though the mRNA is available in the dendrites, it's not translated until there's sufficient stimulation of the spine to allow calcium influx and release from endoplasmic reticulum stores to activate CAM kinase 2, ERK kinase, phosphatidylinositol 3 kinase,